So let's look at the mechanisms of meiosis. And just the same as in mitosis, you have an interphase with a G1, S, and G2 at the beginning. And just like in mitosis, chromosomes replicate in S phase. So we start out with one chromosome here, and during S phase, it becomes one chromosome with two sister chromatids. And here's a diagram of a cell with 4 chromosomes here. So 2n equals 4 in this case. At the end of the S phase, we still have 4 chromosomes, but each one has replicated in size. So in prophase, this is where different things start to happen compared to mitosis. In prophase 1 of meiosis, homologous chromosomes, those are chromosomes that are similar to each other. They physically associate and can actually exchange DNA. And if you see here, we have 4 chromosomes. 2 chromosomes that are similar, that's these two, actually associate and they cross over right here. This is called crossover. And again, another crossover occurring in these homologous chromosomes as well. The fancy word for crossover is chiasma. C-H-I-A-M-S-M-A, chiasma. So if we move on now to metaphase 1 of meiosis, these crossed over chromosomes, still attached, line up on the equator of the cell, or the metaphase plate, but they're slightly different to that seen in mitosis. In mitosis, the chromosomes lined up on top of each other. But if you see here in meiosis 1, in metaphase of meiosis 1, they line up side by side. So homologous chromosomes line up side by side in metaphase 1. In anaphase 1, which was the next step, when they separate, when the kinetochore does its work and they separate the chromosomes, what's happening is you are separating homologous chromosomes. One set goes one direction and the other set goes the other direction. So anaphase 1, separating homologous chromosomes. If you look closely down here, you'll see that the chromosomes have exchanged DNA. There's some blue DNA on this green chromosome and green DNA on this blue chromosome, and ditto for the other combinations as well. And that shows that you are actually exchanging DNA during this crossover process, during this chiasma process. So there are 2 ways of getting genetic variation in meiosis. And crossing over, as we just talked about, is one mechanism. If we start to look at actual chromosomes, then here are 2 chromosomes, one with big A on it and big B, and this one's got little a and little b, so dominant genes and recessive genes. If they do not cross over during the process of meiosis, each of these sister chromatids are separated. If we look down here, you'll see that the little a and little b's go together, the big A's and big B's go together, and nothing really has changed from the original parental cell. However, in the process of crossover, this happens during prophase 1. Here, crossover is occurring right here. This is chiasma, right there. And we'll get crossover and exchange of DNA between these 2 homologous chromosomes, which means now that a big A is associated with a little b, and a little a is associated with a big B, which is a new combination of genes. Follow this through to the result of meiosis, and you end up not only with the parental combinations of genes, big A, big B, and little a, little b, see at the top, big A, big B, little b, but you also end up with a new combination of genes that's big A, little b, and little a, big B, and that's due to the process of crossover in prophase 1 of meiosis. There is a second source of genetic variation, and this diagram represents it happening in metaphase 1. It can happen in metaphase 2 as well. And here we have the chromosomes all lined up with spindle fibers going off to both sides of the cell. And in this particular case, all one type of chromosomes are going one way and all the other type are going the other way. So maybe all the paternal chromosomes are going up and all the maternal chromosomes are going down. But that's purely just a random combination. We could easily swap the positions of this first chromosome. or we could swap the positions of the first and second chromosomes, or just the second chromosomes, all of which, at the end of meiosis, will give you different combinations of maternal and paternal chromosomes. So here we have a quick summary of meiosis. Over here we have prophase 1 with crossover taking place. Nuclear membrane is breaking down. Spindle fibers are beginning to form. This is metaphase 1. Homologous chromosomes are lined up side by side. Spindle fibers are attaching them to the centrioles. The metaphase plate, or the equator, is in the middle. Anaphase 1, we separate homologous chromosomes. So homologous chromosomes, one set goes one way, the one goes the other way. So this whole chromosome goes this way, this top one goes the other way, and here they are separating. Telophase 1, the reverse of prophase, followed by cytokinesis, which splits the cell in two. So the big picture of meiosis is that we have now completed meiosis 1, which is along the first stage here, and we've got to two cells from the division of cytokinesis. There's a cell here and there's a cell here. In between cytokinesis and prophase 2, there is in fact another interphase consisting of G1 and G2. There's no S phase in this second interphase, and so we call this second interphase interkinesis. Interkinesis. No S phase. What would be the problem if we did have an S phase? So now we go on to look at meiosis 2. And you can see meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by cytokinesis. We have two original cells, and so by the time all this ends, we have 4 daughter cells. Prophase 2 is the same in that we lose the nuclear membrane, chromosomes condense, spindle fibers reform. Metaphase 2 is a little different to metaphase 1, but it looks the same as metaphase of mitosis, in that we can see that the chromosomes line up on top of each other here, just line up in a long line, and there are spindle fibers attached to each side of the chromosome, to each sister chromatid. In anaphase 2, seen over here, the kinetochores attach to the spindle fibers and start degrading those spindle fibers so that we split the sister chromatids into 2 separate chromosomes. So this pair of sister chromatids is now split into 2 separate chromosomes, ditto with the other chromosome. Following that, of course, is telophase 2, which will be here, and cytokinesis, which happens after telophase 2. Telophase 2 is just the opposite of prophase again. Reform the nuclear membrane, decondense the chromosomes, and spindle fibers degrade. Cytokinesis, either a cell plate or a cleavage furrow, depending on your species, and you end up with 4 cells that are all haploid and contain half the number of chromosomes as the original parent cell. So if we now compare mitosis and meiosis, both cells here consist of, both parent cells consist of 4 chromosomes, so 2n equals 4. Mitosis replicates, so we now have 8 sister chromatids. We split those cells down the middle, and we end up with 4 chromosomes in each of the daughter cells, genetically identical to the parent cell. This is regular mitosis, just normal cell growth. Meiosis, on the other hand, takes place, and we end up dividing the chromosomes in 2 and then in 2 again, so that the haploid cells produced at the end have half the number of chromosomes. Here we have n equals 2 chromosomes. And if you look, there's 1 dark chromosome and 1 light chromosome. That's one of these 2 dark chromosomes and one of these 2 light chromosomes. So you've picked randomly one of each. So what are these haploid cells that you're making? They are, of course, gametes. And there are 2 types of gametes that you're going to make. These are sperm cells in males and egg cells in females. And if we look at the overall process of gamete production, you can see that in production of sperm, we start out with just a normal, regular spermatogonium, which is actually a stem cell because it because it reproduces again and again and again. And some of those cells then go through the process of meiosis, starting with one cell here, ending up with the 4 haploid cells there. Following meiosis in sperm production, then those cells develop into active spermatozoa. In the human female, it's obviously different. We have an ovum, which is the egg cell, and there are polar bodies that are also made. Now, what does this mean? In the human female, here we have a slightly different process, obviously. Meiosis takes place from here down this diagram, and we have meiosis 1 ending here and then meiosis 2 beginning. But you can see that the products of meiosis 1 are different, and one of these meiosis 1 products then goes on to become the ovum, and the rest of them become something called polar bodies, and in some cases they degenerate. And so although you end up with 4 haploid cells at the end of meiosis, only one of those actually becomes the egg cell. If we look up here in this diagram, we can see that the primary oocyte is arrested in prophase 1. What this means is that the cells go through early meiosis and they get to prophase 1 and stop growing. And this all happens prior to birth. Following puberty and all the way through to menopause, then one of these cells per month is released into the rest of meiosis to produce an egg cell every month. And so some of these primary oocytes can be hanging around for a long time. If they are already present when you're born, and if there's menstruation still happening at, say, age 50, then these primary oocytes can be 50 years old. Can you think of any problems that might occur with oocytes that have been sitting around for 50 years producing problems with chromosomes in the ovum? And that completes chapter 2.